San Marcos. I went to grade them this weekend. I'm like, where are they? So either someone stole them from my car and then everyone gets a 60, or else they're in San Marcos. I'm going to see them tomorrow and then I'll grade them as soon as I can. So we'll see how that works out. So I apologize because everybody wants to see those grades, and I wanted to get it out of the way grading myself, and it just didn't happen. So that's my last chance, so better be there. So that's that. As far as the test, everything else, again, the test is a week from today. It's covering just 14, 15, and 16, not 24. We'll take care of 24 at some point. I want us to concentrate on cardiovascular and blood. The only new material we have, I have to check on one thing, but it's chapter 16, which is going to be very abbreviated compared to most of the other material. You'll see it's pretty straightforward. And the hard material is really on chapters 14 and 15. So we'll have we'll have today and we'll have all of Wednesday, including any lab time, as long as you need me, to go over, review. And what's really critical, if you haven't done it already, although some people I know have because they've been in there, are these extra cardio questions. And we'll talk about a little of those today as well, because we'll do a little of the baroreceptor, depending on our time. The extra cardio questions are critical for this test. Okay, critical. Because I'm introducing things, especially in the second and third pages, which are applications of this, which we haven't really discussed before. And if you don't really understand that, if you don't follow that pattern, you're not going to really be able to get these complicated, critical thinking questions. If you do, and you work with me, etc., during this week, and to go through those, you'll be successful on it. I mean, the test is a difficult test. I gave it a Palomar today, and they gave me one of those looks, when they're walking out. So it's a difficult test. Can you do well on it? Yeah. There's no doubt about it. But it's critical, these extra questions. Now, the answers are online, but it's one of those things that, for some people, if you get it, if you really understand this and you see the answers and it makes sense to you, well, yeah, okay, well, fine. Otherwise, looking at the answers before you try to do this probably won't be to your best advantage. So you really want to study as much as you can quickly, and then at least by Wednesday, although, again, some people have already looked at these, be prepared to really not start when you come in, okay? You really want to look through those, and then we can make sure we review the right things, and we make sure before everyone leaves that you understand that process. Again, the basic idea of that process, and then we'll get rid of today. The basic idea of that process is you're going to put the baroreceptor response on your last page of your test, like, ASAP when you come in probably. Now, not only is that 14 points, but you literally use that, and you'll see this when you look at those extra questions. You can use that to answer 10 more multiple choice questions, talking about sort of this overcompensation, etc. You literally, if you're looking for the answers, they're on your sheet. They're on that question, and they're within my multiple choice, right? And you just, oh, yeah, that's the right answer. So if you understand that concept, it's not just getting the thing down and having it memorized, but if you understand it and understand how to use it, then you'll be successful on this test. I'm positive. Any questions? I did put up. I have those EMGs, finally, that stack up, because I wanted to get some grading in yesterday. I have this morning, I guess. I had the stack of EMGs and the blood typing that you turn in for lab points. You should see those if you were here and turned those in that day. You should see those points up. That adds tiny bits, and there'll be a few others of those that add tiny bits. So chapter 16. Before I do it, what I want to see is there was the last part of chapter 15 has to do with capillary exchange. Did I at least go through that once? Did I go through that at the end last time? Talking about colloid osmotic. Okay, I just want to make sure that I'm starting off with new material here. So chapter 16. Now, okay, not the most exciting chapter, but it's okay. We'll get the basic information down here. So. So let me close the lights. First few slides are really just looking at this. without any particular test questions involved. So we're looking at blood. We have two basic parts of it, the plasma and, of course, the cells. As far as the plasma goes, we already know we're not memorizing this list. But we're going to talk gas exchange for the next test. But you can see, that the liquid part, the non-cellular part of blood, is carrying all of the nutrients and gases, the waste, etc. These, by the way, we talked about proteins being important for proper balance between the plasma compartment and the interstitial. And I said the liver makes these. These are some of the typical plasma proteins we're talking about. The albumins. We'll talk about fibrinogen, which is for clotting. We will talk about that in this chapter. And globulins, antibodies, chapter 24. We'll lay out these. As far as the cells go, this is good news again. So basically, there's three types of cells or parts of cells in the blood. The red blood cells we're going to talk about this chapter. The white blood cells, and you can see them up here, we're going to, since it's chapter 24, and we had that in lab, sort of, I said we were going to revisit it. That's when we'll revisit these, so not for this test. Platelets for clotting are actually, we'll see cell fragments. So this chapter, we'll give a little introduction of how these are made, but basically we'll give you some factoids about red blood cells and clotting and platelets. Overall functions, just to look at, again, we always have this for each system now. Kind of read these. I've already mentioned most of them as well. So chapter 24, as far as the immune system, very important to deliver these. Clotting we're going to talk about now. We talked about thermal regulation, pH regulation, so important. So here's the first thing to really sort of wake up on for a term you might not know, and how we make these cells. So, in fact, we'll come back to this slide, these two slides. But the thing we're going to talk in the next few slides are the fact that all the cell types we're talking about in the blood, the different types of white blood cells, the red blood cells, and these megakaryocytes, which actually are the cell that platelets are derived from, you can see their cell fragments, all of them come from common cells called stem cells, which are in, basically in our bone marrow. So all these cells are constantly, basically, dying. They only last a limited amount of time. So we're going to stimulate the production of all these cells, but they're all coming from this sort of stem cell. And this process is called hematopoiesis, the development of all these cell types from a common stem cell. And so here are the slides about it. I want to talk about what the sort of a mistake the book makes in a second. So where this process takes place to make blood cells, you can kind of see before birth a few places. For us, it's in the bone marrow. Again, cells are sort of short-lived, so we have to constantly replace them. Now, I want to make something which, and I'll put the lights on for this, sort of a pet peeve of mine, because the book sort of makes a mistake. Since I said stem cells, I want to distinguish two types of stem cells. And sort of the differences, and talk about which ones we're talking about here. So there are things which sort of are sometimes referred to as adult stem cells, and there are things which are called, and you may or may not have been exposed to some of this, embryonic stem cells. How many people have heard of embryonic stem cells? Okay, so, and some you may, again, depending upon how much education you've had on this, you might have heard of these adult stem cells too. There's a big difference, and when I teach at San Marcos to non-majors and I throw like 21st century medicine in, I always like people to really understand the difference between these, because a lot of the stuff, since it's controversial, you get misinformation about really what these cells are, what they can be used for, what they can't be used for, etc. And I'm not going to do all of that. But I sort of, since we say it up here, I want to distinguish what these are versus that. Embryonic stem cells, again, are found only, except for some special ways now, are found only in the early developing embryo.
You got a sub?
Okay, it's at one early stage. Adult stem cells are basically found in, basically in most, and we'll say adult, but really it's once you're born, adult tissues. So almost all of our tissues have cells to replace damaged and dying cells. Again, here we're talking about, this is one type of them that are to replace all these blood cells. We know our skin cells are constantly dying. We have adult skin cells that can make skin cells to replace it. In our gut, we sort of are always shedding cells. We have stem cells to do that. Really, every tissue in our body has these stem cells, and they're called adult stem cells. They can be made into these cells to replace them. Now, a term to show the difference between these, besides the where they're found, is has to do with their potency. How potent are they? This is where the book makes one mistake, and at least in this one figure, I changed it. And so, embryonic stem cells. Remember when you were an embryo? Okay, you start off as that one cell, and you're like, what's going on? Divide it to a couple hundred, but you had to become any tissue. And we have 200 types of tissues. So somehow that one type of cell had to become all those types of tissues. We have muscles and skeletal, etc. And that's the potency of these. So they're called pluripotent. These are the ones that are pluripotent, which means they can become all cell types. They have the capability of becoming, under the right conditions, all the cell types that we have in the bodies. So you name a cell in our body, it can become that. Okay? Adult stem cells are basically considered multipotent. So, until we start doing some more manipulation, which we're always doing, the idea is that they can only become certain cell types. So, only become some or certain cell types. I'll put some cell types. Because in one figure here, they might have changed it on another one, but at least on this one, they call these pluripotent, and they're simply not. They're multipotent. The idea is these stem cells that are in our bone marrow are multipotent. They can become white blood cells, red blood cells, and megakaryocytes. They can't become pancreatic cells or brain cells or any of the other cell types that exist in our body, although they're trying to make them become that. But right now they're not, right? So that's the idea. Our skin stem cells can become skin cells. Okay, they can't become heart cells, etc. Versus embryonic stem cells have the potential to become all cell types. And so scientists are working with both these types for cell-based therapy. And there's some controversy on some of these, and there's a lot of information. It's a really cool subject, but not for us. Okay, my only point on this is to distinguish and make sure we understand that when we're talking about hematopoiesis, which is what we're talking about, we are dealing with adult stem cells found in these tissues, which are multipotent and can become, under the right influences, one of these three cell types: white blood cells, megakaryocytes for clotting, and red blood cells. Any questions?
What's the third? Sorry, you said red blood cells, white blood cells.
And these guys. Megakaryocytes,
okay,
which will, fragments from that become platelets for clotting. Are we good on that? So, the next question is, the next question might be, okay, well, these can become all these cell types. What is going to influence this stem cell to become red blood cells versus white blood cells versus platelets? And the short answer is factors. And the table to illustrate this is this. And we're going to want to know two of these. Okay? This last one is so, there's so many factors, it's stupid to sort of memorize it. But the idea is we have two different hormones here. We have erythropoitin, commonly called EPO, and thrombopoietin, called TPO. And so EPO is naturally made kidney. Kidney makes hormones too. It's a really overachiever, and we'll see that. And the idea is when we need red blood cells, since the kidney is seeing a lot of blood, okay, going around, it's going to produce the EPO. Yeah, sneeze out chalk. Whoa. And then influence that process to become red blood cells. Liver makes thrombopoietin, or TPO, that's going to influence it to become megakaryocytes for clotting. So when we need clotting factors more than others, we're going to cause it to shift that way. White blood cells, as you can see, a bunch of stuff, okay, that we're not going to go into for this course. So there's no point in remembering that. But these have common names, and so you sort of want to remember this. And most people, how many people have heard of EPO? Okay, yeah, you should have heard of EPO, especially if you plan on cheating in bicycle events for a decade. You know EPO really well. Okay. So again, EPO will burst red blood cells. There is synthetic EPO that if you've had, let's say, chemotherapy and you're anemic or even otherwise, they can give you synthetic EPO to stimulate your production of red blood cells. Athletes use it to try to boost up their red blood cells so they can carry more oxygen, and we know all about that. But this is what you need to know from that overall. EPO and TPO to directed toward red blood cells are megakaryocytes. Speaking of hematocrits that we measured, so out of all this, again, sort of a blood panel here that we're not memorizing. You kind of see some of this stuff. But the idea is that what hematocrit is what I want us to know and sort of the importance of it. Hematocrit, as we said in lab, is measuring out of your whole blood what percentage is occupied by red blood cells. And it's one of the factors that can determine whether you're anemic or not. We also look at hemoglobin levels, etc. And you can see, like we did in lab, although you're not memorizing, males have generally higher and higher average, but there's a huge range in individuals as far as what a natural hematocrit is. Okay, that's hematocrit. And again, we're going to relate this to something called polycythemia in a minute, which we have too many red blood cells. But that's what you need to know from this, so you're not memorizing this. So, some factoids about red blood cells. And I'm going slow because then I got some how to clot, and then we're done. But we're not really done. But we're done with the new material. So, as I said, this chapter is just, a little icing. Okay, so what about red blood cells, Mike? Make them exciting. I can't. Okay, they contain no nucleus or organelles. So if you sort of look at their development, they're very specialized. After this test, so next Wednesday, we're going to definitely start talking about the respiratory system. It is designed to efficiently have hemoglobin to carry oxygen and also CO2. So hemoglobin is sort of it. So it gets rid of organelles, nucleus, and basically it's hemoglobin and all the enzymes needed to get this thing working. And so that's sort of why we make room for it. Now, since it doesn't have mitochondria, that's an organelle, it means it's basically going to use sort of that anaerobic respiration. It's not using oxygen, it's using those first few steps called glycolysis. Like when we're sprinting, we use a lot of glycolysis. So that's what the red blood cells have to use. So, point. And all this, it sounds good. I should make these slides up and put pretty pictures because it's like a lot of words. And all this says is stuff we already know, which is that the red blood cells have a cytoskeleton and they're going to respond to osmotic pressure. Osmotic pressure still stays big. This is a normal cell. This is sitting in what? What type of solution is this? How many people say hypotonic? How many people say hypertonic? Water goes to the party, crenate. This is in a hypotonic environment, lower goes in. Hemoglobin, a brief introduction. Here. So, again, next week we deal with hemoglobin a lot. Just an introduction to it, though. It's, since red blood cells, that's its main component. It has 4 protein chains, okay, 2 alpha, 2 beta. And each one has an iron-containing heme group that can carry oxygen. So this can carry 4 molecules of oxygen. Much more to come next test. And one Friday night, I downloaded this picture of hemoglobin, and I was just like, wow, that's cool. So some people study the structure of everything, crystallographers, and they're really, this looks cool. Anyway. Yes?
That's hemoglobin. What's surrounding hemoglobin?
What does that mean? From this picture or this picture?
Or that one, yeah. The second one.
This?
It's just inside the cell of the cell.
Well, the red blood cell, yeah, surrounding. I mean, there's thousands of hemoglobin molecules contained within a red blood cell. So the red blood cell is surrounding this,
Okay.
and we're going to show you oxygen's going to come in. So we're going to have diffusion through the red blood cell.
Are those protein or the enzymes rather?
Say it again.
Are these all things the other enzymes that are with it?
Yeah, you can't see them there. Other enzymes are with it, right? All this is like just the crystallography. You can actually see the alpha helical part of the chains, and there's the iron-containing ring. I didn't mean to get people interested in this. I thought that would be a biochemistry course. But we care about bilirubin. So since red blood cells don't last a long time, we've got to remove them because they contain that iron, which can be toxic in some products. So we're going to convert hemoglobin to a compound called bilirubin. And the kidney I just call it the kidney because they work together. The kidney and liver will basically metabolize these to make them less toxic, and we'll get rid of it. So bilirubin, a byproduct, and we'll get rid of hemoglobin. If we have too much bilirubin, jaundice. So jaundice is the buildup of too much bilirubin. And we'll talk a little more about that in the respiratory system too. Yeah, this is my little happy face. See, I can tell when I was making this up, I had to entertain myself. So I'm looking for cartoons and pictures. All right. So we know what jaundice is. Yes? Check. Okay. Problems. Now there's anemias, and to be honest, we won't be testing much on anemias because we already know what it is. Basically, different types of anemia, we don't have functional hemoglobin. We don't have enough red blood cells, we don't have enough iron, or we have some sort of mutation in which the cells don't work right. There's all kinds of different anemias. And I don't feel like memorizing, or which one is which, but it sort of makes sense. Since iron is necessary for carrying oxygen, that might be a deficiency, you might need iron. But also just other anemias, you don't have enough red blood cells themselves, you won't be able to carry oxygen as well. So that's anemias. But this one you might not have heard of before, and so this one is more what I'm concerned about, even on the extra question. So the term polycythemia is too many red blood cells. Okay? So when you're looking at a hematocrit, you're talking about, if you have a high hematocrit, higher than normal. Again, remember we have the two areas here. But if we have, an increase in red blood cells over the total, so now we have, that's too much, have about 6 out of 10. Then you might have a hematocrit of 60%. Well, that's indicating polycythemia. And remember, the idea of polycythemia, or one idea of this, besides maybe you can use it to your advantage at a sporting event, but the other idea is when we talked way back at the beginning of the section, we talked about things which control resistance of flow. Okay? And one had to do with vasodilation and constriction, the other had to do with viscosity. Now we mentioned that if you have a high hematocrit, it's going to be more viscous, and that means harder for the heart to pump. And so, polycythemia could definitely be one way that happens. And so, if you look at polycythemia, there's a couple ways you can have it, obviously, but one term I do want you to know, a subclass is relative polycythemia. That was the idea of if you're dehydrated, you have less plasma volume, you haven't lost your red blood cells yet, and that can give you a high hematocrit polycythemia as well. So, you can get EPO and have too many red blood cells, that's the type of polycythemia, or relative polycythemia, you're dehydrated, you've lost plasma volume, therefore your hematocrit is raised, and you can have some of the same symptomology. So, that term I needed to know. Polycythemia, and specifically also relative polycythemia.
And if we increase resistance, do we increase blood pressure?
Ultimately we'll see, but yes. The idea is it's going to create. Well, actually, I shouldn't say that. I shouldn't say that because I can't know that for sure. You're going to increase, if you increase resistance, you have more resistance to flow, so your heart's going to have to pump harder to do it. But I don't really know ultimately if that's going to change the blood pressure or not. I just don't, I have to think about that more or look it up because, doesn't necessarily, it just might compensate to push harder. Whether it creates more resistance, as far as more pressure here, I'm not sure. It definitely is going to, decrease flow or interfere with flow and making cardiac output go up. Good question. If I can remember to find out, I will. We're to clotting. Okay, that's about it. That's it. I was like, hey, I didn't like this chapter though. Let's go into the other one. So, this is what we need about platelets and clotting. We'll literally be done at 6, and then we can deal with other matters. Start reviewing. Okay, dot. We got these bullet points. Now the idea of these two will be that when they're inactive, you can see their cell fragments, they're not going to be very sticky. They're not going to be sticking to each other. They're basically, and we'll show you why, we don't want them to come together and start to, get this reaction going. But to fit it into this and to actually have a process, which is a little more exciting, this is how we do this chapter, to finish it up. There is the process hemostasis. We've damaged a vessel. We don't want blood to leak out. We want the blood to stay where it is, hemostasis. And so we have 3 basic steps. We're going to first produce local factors that will cause vasoconstriction. So we have less blood flowing through this damaged region. Okay, makes sense. Then the idea of the platelets now. We're going to form a temporary fix, and we'll show a nice figure which shows that, called a platelet plug, which will be very quick. And then we're going to form a clot, which will be more longer lasting things. 1, 2. So I've already mentioned this. We're not going to talk specifically about the factors, but we're going to be basically, factors are going to be released to cause vasoconstriction, to limit flow. So, here's a figure. Finally, this is a good figure. We're talking about how to form a platelet plug. Do I have, okay. So, the first thing actually I put up here, and again, we're going to pretty much follow this. I have a couple of details that I put up on the top here. But the first one I want to put here is actually what I call step 0, that the platelets themselves, we don't want to start adhering and forming, platelet plug and clotting unnecessarily. So the endothelials themselves basically release substances, prostacyclin and NO, which is not NO, it's nitric oxide. So this is before there's injury. So with no injury, the endothelial cells produce these substances to help prevent platelets from coming together and starting a reaction. So, but here now we've had damage. And so upon damage, again, we're going to sort of see, we're exposing underlying tissue, including collagen, and some of these platelets will bind to it and deactivate. Pretty much what that says. Then they're going to basically release factors to try to attract other platelets. And these factors I just put up here. So these are the factors that are being released, or 2 of them that I want us to know. So once platelets get activated and they start sticking to this, they release ADP and thromboxane A2 that will attract other platelets and basically start forming this platelet plug here. So ADP and thromboxane A2. And that gets more platelets, and we have the plug. That's simple. I know for you guys it's like, come on, but I'm sorry, this is all. So let's have an easy chapter. Let's play with the plug. Just put that whole picture together one more time. We only got like 2 more slides. This is like scary. I'm trying to slow down. Maybe I just speed up and get over it. Okay, here's play with the plug. So any questions about this though? Again, we have these substances that prevent platelet adhesion when we don't want it. Then we're going to have damage, platelets basically bind to the collagen, become activated, release ADP, what is this, and thromboxane 2 to attract more platelets, and we form a plug or a temporary fix. Very quick.
Is this something you want us to memorize step by step?
Well, I don't call anything memorizing. Do you want to know the steps of platelet plug formation? Sure. Okay, blood clotting. Here's our last step. And what we're going to see is something, well, maybe I didn't show it before. We'll see on my thing. We're going to be seeing an enzyme cascade, and I have to talk about Zymogens. So you have this, but I'm going to show this here. And so when you look at this, and I'm going to spend a couple, since this is only, we only have 2 pictures left, I want to show this. So this is the clotting cascade. And there'll be a 3-point essay question to memorize all this, and no, I'm not. I never even had to memorize all this. We're going to just look at these last couple of steps. But the idea of this pathway, and we'll put these factoids up there, is whether I mentioned this before, because we skipped some chapters, I don't know, is, I think we did though. Did I ever talk about Zymogens? I was jumping up and down. Zymogens. Okay, well, let's review what they are. So there are things which are, because we're going to see this next unit. So Zymogens are basically inactive proteins. They sort of have, I don't know if I have my colors there. No, really, I do. They basically have sort of a protein with sort of an additional piece to it that makes it inactive. Okay? Then we cleave that piece to become an active protein or enzyme, but an active protein by clipping this, and now it's active. Now and again, we're only going to look at the bottom ones to help recognize it. 2 of the ways we recognize by name Zymogens versus their active form has to do with putting a pro on front. So prothrombin is inactive, becomes thrombin, which becomes active. And the other way, and these are the only ones you have to know anyway, the other ones are having something like an ogen. So fibrinogen is inactive. It's going to get converted to fibrin to be active. Okay, and then we're going to cross them. And so all of these are Zymogens. And this is a Zymogen cascade. And this is why, luckily I never had to memorize the order of these factors, because they're just called them factors: inactive, active, inactive, active. All of these represent a Zymogen cascade, and where once something triggers, including damage, when something triggers the cascade, you're going to have the first one, Zymogen becoming active, that activates the next one, activates the next one. This is a positive feedback loop, okay? Because we want this to happen very quickly. Again, the advantages of Zymogens are the fact that we want to make sure that everything there is ready to go. We don't want the liver, to tell the liver, let's start making clotting factors on bleeding to death. We want them there, but we want them inactive until we're ready. So something's going to trigger it, and this will happen very, very rapidly. So again, the factoids of this, liver does make these, because we care a lot about liver starting to become important. We need vitamin K to make these. And if you look at every step, calcium is the big star of this whole section. At every step, basically, pretty much every one, you need calcium as a cofactor to make the enzyme work. That's why we're seeing calcium being important for clotting.
When you say to make enzyme work, you mean become inactive to active, right?
Yeah.
Which ones are called Zymogens? I thought you said you're getting progens and it's inactive. The Zymogens are the inactive protein.
Yeah.
So you're saying that what are thrombin and fibrin?
So prothrombin, that's good, so I can put it up here too. Prothrombin is the Zymogen, thrombin is the active form.
So there's not a name for the active form, it's just active protein. That's what I was saying. 'Cause I thought you said all of them were Zymogens, but only the inactive ones are, right?
I don't know what that means. Every one of these has every one of those things has 2 forms. An inactive form and a active form. Okay, and then for these last 2 sets they do have specific names where prothrombin is the Zymogen of thrombin, and I'm probably confused on what you're asking, let me just write this too. Fibrinogen is the inactive form of fibrin.
So thrombin and fibrin are the enzymes.
They're going to be the active enzymes. Okay, these are the inactive proteins or inactive enzymes that when we're ready, we do. You can see the previous one. We'll activate them, clip off that piece. Again, basically, it actually acts as the clipper itself once it becomes activated. So it basically will clip off the piece, and now we have the active form. So this is just the active form of the zymogen for both of these. And so all of these other ones are the same way. There's an inactive factor 12 or whatever, an active one. So they're just being converted by clipping off that piece.
And are you also stating that they're all connected, like thrombin becomes fibrinogen?
No, I'm saying, and we'll see this, we'll just look at this particular part, that, first of all, something else, again, we're not memorizing any of this anyway. Prothrombin is going to be activated to thrombin, okay? The zymogen is going to be activated, and now we have the active thrombin. Once we have the active thrombin, it is what's going to convert fibrinogen to fibrin. Each one of these becomes sort of the one to the next one. Basically, it will be the one that clips off the next guy. So, then the only ones we need to know are these last ones, even though that's happening all there too. So, if something else is going to activate a prothrombin, then it's going to be activated and become thrombin. Thrombin now is active. It is going to convert fibrinogen to fibrin, and basically that's going to form the clot. That's going to be fibrin. Okay? And that's the part of the clotting cascade. I mean, I have one more slide to show. But that's as far as the clot formation. Again, it's not memorizing all this, but basically these last couple steps, because they have names, are sort of the important ones. Prothrombin to thrombin, fibrinogen to fibrin. When we have a clot, do we want it to stay there forever and ever and ever? No. All right. So breaking up the clot. Well, first of all, let's just look at this, and then we'll show the two ways to do it. So one of our best enzymes for clot breaking is called plasmin, and guess what? It came from a zymogen. So it's the same idea. We make the inactive form, and when we're ready, we convert to the active form. So the inactive form is plasminogen. That is the inactive, or zymogen. We can activate it to become plasmin, and that breaks up the clot. It starts eating up the fiber. But two sort of ways of diagramming here. One interesting sort of weird way is that thrombin itself, remember we just had this we're going to memorize. Thrombin converts fibrinogen to fibrin to form the clot. It basically slowly, and I don't know exactly how slow, but somehow slowly activates, converts plasminogen to plasmin. So it's sort of planting the seed slowly of its own destruction. But I don't know, people are like, well, how much? I have to look up there to see how quickly. Its main action, quick action, is to form the clot. But this one you might have heard of, so let's know this one. So TPA, tissue plasminogen activator. Actually made by the liver. And you put that up there, that's liver. But that will convert plasminogen to plasmin to help break up clots. Now, if you've got a clotting problem, they can give you TPA, and that might help break up the clot, but you might bleed to death. So it's up to you, because obviously it's going to prevent your ability to form clots effectively too. So if you have some kind of bleeding going on that you need to form a clot, that could be a problem. So I think that's pretty critical when they try to figure that out. But potentially it's a clot breaker, because again, TPA converts plasminogen to plasmin, plasmin breaks up clots.
Is that involved in hemophilia, or is that?
No, hemophilia, just since you just asked, is, I don't remember which one. It's a genetic disease in which you're missing one of those guys, factor 7, factor 8, something like that. And so then your inability to form a clot.
So just by one, it can mess up the whole chain.
Yeah, I mean, just think of dominoes, and if you sort of mess up one of the dominoes, it won't do it. You need, each one is necessary to activate the next, to activate the next. Yes?
Just out of curiosity, is a scab?
Well, scab's not really a clot.
It's not like when you
No. It's what we call a scab. You have to. Like I said, short chapter. So again, it's up to you. I'm going to take a few minutes this week because some people might want to leave. Okay, I mean, all I'm going to do is, I don't know, we'll go over, we'll practice some things, review some things, start looking at the extra questions maybe. Again, all we have left this week is that. So it's up to you how prepared you are, what else you got going on in your life. I have nothing new to say, but we're going to do a lot of review over the next, day and a half or something like that. So.
Sorry, I missed the first minute or so. Is next Wednesday, are we doing this review as well like we usually do?
This Wednesday, yeah.
Okay.
That's why we just have a lot of time to review this week. But especially the extra questions. If you haven't looked at those yet, definitely by Wednesday have to look at them and try them, although we might talk about a little of them today too, so we still have 45 minutes. Again, the lab itself, who's in lab tonight? I've abbreviated that a lot too, so it won't be, we literally are going to do our EKG, and talk about respiration's effect upon EKG. So I don't know. I mean, I could spend what is that, 45 minutes? It's under an hour for sure. And so then I'm also available to go over material as well. And so it's just this week. Again, this is a hard test. Okay, I've got a lot of smart people in Palomar who had a headache. Okay, and I looked at it. I mean, some of them did okay, but they're not as good as they usually have done. Okay, so I want you guys to do better. So even you A students, okay, you want to know this material really well, so to do well on the test. And everybody else, don't fear. We got all this time. Do not fear. You can do it, but it is a difficult test. So take a few minutes, see what you're going to do. Take a break, leave, come back, and. We'll do pretty much what you want.
But it looked like an exciting game though. I mean, how many pieces on the show? I saw the beginning.